Hello and happy Tuesday. Today's class, we spent a little time reviewing what caused the sickle cell trait. And then I wanted to know if you know what the difference between sickle cell trait and sickle cell disease is. So we talked a little bit about these three guys right here. Ryan Clark is the player who, in a game against the Broncos, he played for the Steelers. He had some blood clots and he lost his spleen and I believe a kidney because he has the sickle cell trait. Tevin Coleman and John Brown are two other players that in the NFL who both have the sickle cell trait. Um, and go ahead and pause the video and see if you can remember what causes that trait. And then when you unpause it, I'm going to go ahead and talk. So what causes the sickle cell trait is, at its most basic level, a mutation. A mutation, as a reminder, is a change in the DNA sequence. So it goes from CTC to CAC, or in the case of sickle cell anemia, you go from GAG to GTG. Oops, sorry about that. From GAG to GTG, which means you're going to be coding, instead of for glutamic acid, for valine. So now you've got a different amino acid sequence, which means that your protein is going to be a little bit different shape and it's not going to work as well at grabbing oxygen, and it's going to cause more blood clots because those sickle cells, blood cells, kind of get caught up in your bloodstream. So that is what causes sickle cell anemia and sickle cell trait. And what causes sickle cell disease, go ahead and pause it if you want to try to answer that. And then when you unpause it, the difference between sickle cell disease and sickle cell anemia has to do with this picture right here. So as a reminder, you get 23 chromosomes from your mom and 23 from your dad. And let's say that your dad has sickle cell trait and he gives you that chromosome, but your mom doesn't. And so now you have one good and one bad sickle cell trait, or one good and one bad hemoglobin gene. Overall, your body still has one good copy of the gene, so it knows how to make good hemoglobin. So we would say you have the trait, but you don't have the disease. Now, if on your dad's chromosome and your mom's chromosome, so let's say they each gave you the bad version, the sickle cell trait, now you've got two bad versions of the hemoglobin gene, and so your body is not making hemoglobin correctly. Now you have sickle cell anemia. So that's the difference between just having the trait and having the disease, is whether you have one copy or two bad copies of that gene. So that is the answer to that question. And now what we're going to go to is that worksheet that I gave you all before you left, the mutations worksheet. So what you should do, get that sheet out, and the very first thing is you should try to transcribe the DNA into mRNA. That means taking that DNA sequence and putting the corresponding RNA base across from it. I'm going to show you how to do that in a second. The second thing you can do is take that mRNA sequence that you made and turn it into amino acids. And then the final thing you're going to do is there are four mutation sequences, or four mutated sequences. There's a silent mutation, there's a missense, nonsense, and a frame shift mutation. So I'm going to go ahead and walk through that really quick. So let's see. Okay, so this is what your sheet should kind of look like. And you've got this sequence up here that is the kind of original DNA sequence, or you could look at this right here. This is what I gave students in our class today, a kind of a bigger version that's easier to write underneath. And where you see a C, you should put a G. Where you see an A, you should put a U. Where you see a G, you put a C, and so on. So you should be, you should be writing that code, and then every three bases you can put a line. And then that would be G, G, C, and then we'll put our line. The reason we're putting our line is that it represents the codons. It represents the idea that the ribosome is going to read this mRNA three bases at a time. So then we've got, after that, we've got G, U, C, and you can just keep on going all the way down the line. And then what you're going to do after that is you're going to open up your codon chart. And what you'll do, you see GUC, and on here, GUC codes for valine. So now below here, I would write valine, and then GGC, oops, sorry. So my next codon is GGC, and we look up GGC, that is glycine, or GLY. So that is GLY, and so on. So you're going to go through this whole thing, coding out all of those amino acids. And then now back on the worksheet here, I have made a change to these four sequences. There's just one change in each one of the sequences. You don't have to necessarily transcribe all of these, although you can do that and it'll help you. But what you need to do is find the mutation and see what it changes about the amino acid sequence. Each one changes the amino acid sequence in a little bit different way. And I recommend checking in with Dr. Hill if you are stuck on this, because I know that he can help you. And so can Ms. Newman. But I want you to see if you can figure out what the difference between a silent, a missense, a nonsense, and a frame shift mutation is. Go ahead and pause the video before you work on the worksheet, and I'm going to help you with the answers a bit. Okay, so if you want to hear the answers now, you can unpause it. I'm going to go to the four mutation types. A silent mutation means that you have a change from TTA to TTG, but they still code for the same amino acid, leucine and leucine. So it's silent because, yes, it is a change in your DNA sequence, but it doesn't actually change the amino acid sequence, so it doesn't really cause any major problems in your body. A missense mutation means that TTA, I'm going to get my pointer here, hold on. TTA gets changed to TCA, and instead of coding for leucine, now you code for serine. And that's a problem. That's what causes sickle cell anemia. Remember, was that change from glutamic acid to, I don't remember which one that changed to off the top of my head. And then the third kind, the nonsense mutation, this is a really bad mutation, is when the sequence gets changed from TTA to TGA, which is going to now code for stop. If you look that up on your codon wheel, what that means is it tells that ribosome to stop coding. So we go back to this guy right here. As that ribosome is going along and reading your mRNA sequence, when it sees stop, it just stops building that protein, which means now your protein is going to be really small and not complete. That's a bad mutation. Another one just as bad as a nonsense is the frame shift mutation. And what happens with that one, notice that C, G, T, 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 now we're missing a G, but then A, C, 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 T, T, A. Everything else is still the same. Basically what happened was one of these bases got deleted. In this case, it was the A was removed from there. Wait, no, sorry, it was one of the T's. So in a deletion, you remove a base pair, whereas in a you can also have an insertion, so a base pair gets put into there. But basically, it's going to slide everything down. So instead of reading TTA, now it's going to read TAC, which codes for a different amino acid. In this case, that codes for tyrosine, and then you get leucine, and then you could get stop, or you could get a whole bunch of wrong amino acids. So basically, a frame shift makes everything slide down by one, which means now your frame that the ribosome is reading is wrong. So that's really bad. And then the last thing we talked about in class today was, in preparation for talking about chromosomal disorders tomorrow, is we spend a little time talking about the four main classes of disease. That's infectious, deficiency, cancer, and congenital. So maybe pause and take a second to talk with somebody near you, or just think about if you know what each of these types of diseases are and maybe an example. So go ahead and pause it. And then I'll go ahead and talk through this real quick. An infectious disease means you can be infected by it. It means you can catch it from somebody else. Typically, infectious diseases are caused by a virus or a bacteria getting into your body and causing some problems inside of there. Examples would be things like the flu, the common cold, Ebola, HIV, which causes AIDS, chickenpox, lots of different kinds of viruses that you can catch from outside your body that are now causing problems inside your body. And then there's deficiencies. A deficiency means you don't have enough of something. A common example of that is an iron deficiency. It's known as just anemia or scurvy, which pirates get or used to get, is when you don't get enough vitamin A. There's lots of different things that can cause that can happen when you don't get enough of some vitamin or mineral in your diet. And then cancer. Cancer is a very specific type of mutation that you get in the course of your lifetime. So you're not born with cancer, but you can acquire cancer, and it's a mutation in the DNA that causes mitosis. So when mitosis is supposed to start or when mitosis is supposed to stop, you've got genes that tell your body, those cells to either do mitosis or not do mitosis. And when you get a mutation in both of those, mitosis kind of gets stuck on, so that the gas pedal that tells your cells to go gets stuck on, and the brake pedal that tells your cells to stop is broken, so you can't use it. Your cells just keep on splitting and splitting and splitting and splitting and splitting and splitting, and eventually now you've got a big mass of cells that we call a tumor. And then the last one is a congenital or hereditary disease. And this is a little bit of a mishmash of a category. It's basically something that you are born with. There are some congenital diseases that you maybe get in utero, so maybe there's something that happens when you are in the womb. Something like fetal alcohol syndrome would be considered a congenital disease, but you didn't get that because of your parents' genes. Mainly I want to focus on hereditary, and a hereditary disease is a disease that you get from your parents because they had it and you inherit it. You get it in a genetic form from them. So that's what we're going to be talking a little bit about tomorrow in terms of chromosomal conditions like Down syndrome and Edwards syndrome and Patau syndrome and Turner syndrome. We're going to be talking about how those come about. That is it. I will talk to you tomorrow.